Hi children, I am R. Sunitha, School Assistant Bioscience, ZPHS Nerada, Mandal Kuravi. Today I am going to teach about cell division, mitosis. Continuation of life begins from the cells. Those are either vegetative cells like general body cells or sex cells like gametes. Rudolf Virchow, a proponent of cell theory, is given a credit for the phrase omnicellula decellula. It means cells arise from the pre-existing cells, indicates the importance of cell division in the creation of new life. In 1852, a German scientist, Robert Remak published his observations on cell division based on his observations of embryos. This was one of the first attempts to understand the mechanism of the cell division. He stated that binary fission of cells was means of the reproduction of the animal cells, and he also stated that what happens during cell division could only be understood better when scientists came to know what is present inside the nucleus of the cells. In 1879, scientist Walter Fleming examined many kinds of plant and animal cells, and he selected those that showed division, and he reported his observations of such cells that there were string-like structures in the nucleus which are split longitudinally. In the process of division, he named such a process of division as mitosis. Here mitos means fine threads, as the dividing structures resemble as fine threads. Walter Fleming made meticulous observation and sketches. He observed that there were a sequence of events occurs in a cell division. A decade later, those dividing structures are named as chromosomes, means colored bodies. The most important discovery of Walter Fleming was chromosomes appear double in nature. And Wilhelm Raux proposed that chromosomes carried a different set of heritable elements, and the longitudinal splitting proposed by Fleming ensured that the equal division of these elements. In cell division, the cell divides into two halves with the equal number of chromosomes which are similar to the parent cell and diploid in nature. August Weismann, a biologist, hypothesized that in a successive generations, individuals of the same species have the same number of the chromosomes. In a successive cell division, the number of chromosomes always remains constant. The cell division in the vegetative cells is called mitosis, which completed in 40 to 60 minutes. The scheme of mitotic division was confirmed in 1904 by Theodore Boveri. The period between two mitotic divisions, two cell divisions, is called as interphase. This interphase can be divided into three phases: G1 phase, S phase and G2 phase. And mitotic division, the mitotic division occurs in two phases like karyokinesis and cytokinesis. Now we will discuss about interphase. This interphase can be divided into three phases like G1 phase, S phase and G2 phase. The G1 phase is a linking period between the completion of the mitotic division and the DNA replication. In the G1 phase, the cell increases in the size, and the second phase of the interphase is S phase. This is a phase of DNA synthesis or DNA replication, and the third phase is G2 phase. This G2 phase is linking period between the DNA replication and the beginning of the mitotic division. In G2 phase, all cell organelles divides and chromosomes prepares for the mitosis. So after completion of the interphase, there is a mitotic division, there is a mitosis. Mitotic division occurs in two phases like karyokinesis and cytokinesis. Karyokinesis is a division of nucleus and cytokinesis is a division of cytoplasm. So in karyokinesis there are four phases, those are prophase, metaphase, anaphase and telophase. In prophase, the chromosomes contracts, spiral, become visible, and chromosome splits lengthwise, form chromatids. These chromatids connected by a centromere. In prophase, the nuclear membrane breaks down. Centrosomes, which having the rod-like centrioles, divides to form ends of spindle fibers. Perhaps this is occurs in animal cells. In metaphase, chromosomes moves to the center, moves to the equator of the spindle. These chromatids attaches to the spindle, centromeres attaches to the spindle fiber, and these centrioles splits and separating the chromatids. Third one is anaphase. In anaphase, the spindle fibers contracts, pull the centromeres, which attaches to the centromeres, pulls the chromatids towards the poles. Pulls the chromatids towards the poles. In telophase, the chromatids in telophase, chromatids elongated, and again the nuclear membrane formed around two daughter nuclei. So this is about karyokinesis. And cytokinesis occurs after the completion of the karyokinesis. In cytokinesis, cell membrane in animal cells and cell wall in plant cells pinches into form two daughter cells. So this is cytokinesis. So after the completion of the karyokinesis and cytokinesis, here two cells are formed from one cell, which cells are similar to the parent cell, which cells are identical copies of the parental cell. So this is about the mitosis. Thank you.